ASRB net preparation model paper 4 on especially on bacteriology. I'm Anand Shankar Reddy, PhD plant pathology. First one is first plant bacterial disease was discovered in the year. So first plant bacterial disease was discovered in the year 1878. So that was first discovered by T.J. Burrill. There is a scientist called T.J. Burrill who reported the first plant bacterial disease in 1878. That is a fire blight of apple and pear. But the first proof was given in the 1882 only. So the answer is 1882. Okay. So if you see in the G.N. Agrios book also it is mentioned as 1878. In 1878 he have an idea that if he's doing right, that is the, I mean, he mentioned in one of the paper, if I am doing right, and plant bacteria also can causes the plant diseases, and later he started working on it, that is fire blight of apple, and he identified with proof. The proof was given in 1882, so the answer is 1882. Make sure the first plant bacterial disease was reported in there 1882. You have an idea in 1878, but was the proof was given in 1882. So that was T.J. Burrill. The disease is fire blight of apple and pear. Fourth Koch postulate was given by. So there are four Koch postulates. First three Koch postulates was given by Robert Koch, and fourth Koch postulate was given by E.F. Smith. E.F. Smith is generally considered as father of plant bacteriology, but first three was given by Robert Koch only. Stanley Falco, he gave molecular Koch postulates in 1988. Louis Pasteur, we know variable pasteurization, germ theory of disease and all those things. And first plant bacterial disease, that is fire blight of apple, is caused by Irvinia amylovora. Irvinia amylovora, but before naming this Irvinia amylovora, it is called as Micrococcus amylovorus. This question was asked in 2021. Fire blight of apple is caused by Irvinia amylovora, but they have given what is the primary name of fire blight of apple? Micrococcus amylovorus, later name changed to Irvinia amylovora, probably in 1920. Father of plant bacteriology, just now I told, right? E.F. Smith. E.F. Smith is considered as father of plant bacteriology. Penicillin, it is actually antibiotic, you guys know very well, right? So penicillin was first discovered by Alexander Fleming. Alexander Fleming discovered the world's first antibiotic in 1928 around September time, and he published the article in 1929 around August time. In a couple of books it is mentioned that 1929. No, it is discovered in 1928 itself, but it took a little bit time to publish. But discovery year is 1928 only. So, and he got Nobel Prize along with Flore and Chain in 1945 for the mass multiplication. These two persons got for mass multiplication. Alexander Fleming got for the discovery. Streptomycin was discovered by. Streptomycin was world's first broad spectrum antibiotic. So world's first antibiotic is penicillin only, but world's first broad spectrum antibiotic is streptomycin, that was first discovered by Selman Waxman in 1944. Tundu disease of wheat is caused by. Tundu disease of wheat is caused by. It is actually associated with a nematode disease. Generally, it is caused by Corynebacterium tritici. Generally a bacterial disease associated with a nematode called Angina tritici. Angina tritici and Corynebacterium tritici. It is an association. So this is the answer. Hypersensitive response in bacteria was discovered by. So the first hypersensitive response when it comes to bacteria was actually discovered by Clement, probably in 1963, I think so. Antifungal antibiotic Hamycin and thermostatin. It's not thermostatin, probably it's a dermostatin, I think so. It was first given by a father of Indian antibiotics, that is M.J. Thirumulachar. So M.J. Thirumulachar was well known for discovery of first antifungal antibiotic Ariofungin, and Hamycin, dermostatin, SYC4 and all those things was given by Thirumulachar only. First bacterial avirulent gene was discovered by. See, what we have seen is a hypersensitive response, that is Clement. Now they are asking is the first bacterial avirulent gene they are asking. So the first bacterial avirulent gene discovered by is Staskavix, probably 1984, I think. Staskavix. And the size of the bacteria, generally bacteria sizes ranges from 0.5 to 1 into 2 to 5 micrometer in diameter. So the answer is C, I think. Yeah, 0.5 to 1 into 2 to 5 micrometer in diameter. Budding bacterial genus, generally the spores which are produced through budding is known as blastospore, right? Blastospores. So budding, majority of the bacteria can reproduce by means of binary fission. Couple of bacteria can also reproduce by means of budding. So such kind of bacteria, general example they are asking, rhodomicrobium is the example. And the shape of the Dello vibrio, generally rod-shaped bacteria or maybe such kind of things will be there. In morphology, we might have studied in the previously published videos. The shape of the Dello vibrio is a comma shaped. So comma shaped bacterium, Dello vibrio. Dello vibrio generally look like a comma shape, like this, comma shaped. Give an example of a kidney shaped bacteria. So which bacterium look like a kidney shaped bacterium? Corynebacterium look like a kidney shaped. Next question is the double stranded DNA structure of bacteria. So which of the following which is a double stranded DNA structure which is present in the bacteria? Plasmid, cytoplasmic organelles, ribosomes, intercellular. So we know very well this plasmid is a double stranded structure, like it is extra chromosomal circular double stranded DNA which is present in the bacteria, right? So bacterial cell wall is made up of, we know very well peptidoglycan, we can also call as mucopeptide, right? So what is the composition of peptidoglycan? It is a combination of NAG and NAM. NAG mean N-acetylglucosamine and N-acetamyl muramic acid constitute to form beta 1, 3 or maybe probably 4 linkage, right? So composition of peptidoglycan, this linkage is also important. I think in fungi it's 1, 4, I think so. Maybe in bacteria, no, in bacteria it's 1, 4. NAG plus NAM constitute from beta 1, 4 linkage. Okay. The next question is composition of peptidoglycan. Just now we discussed NAG plus NAM constitute to form beta 1, 4 linkage. Ralstonia solanacearum race 4 will, in fact there are 4 races are there: race 1, race 2, race 3, race 4. Generally this 5 races are there: race 1, race 2, race 3, race 4, race 5. They have asked is right, race 4, right? Race 4 generally known to infect a ginger, race 5 mulberry. So race 1 solanaceae, race 2 musaceae, race 3 potato, race 4 ginger, race 5 mulberry. I think these are all the 5. So the 19th one is almond leaf scorch is caused by Xylella fastidiosa. There are a couple of other diseases like peach leaf curl, like Pierce disease of grapevine, almond leaf scorch, citrus variegated chlorosis, alfalfa dwarf, such kind of disease all are known to be caused by Xylella fastidiosa. And this is also known to be the first plant pathogenic fungus to be completely genome sequenced. Scientific name of glassy winged sharpshooter. This Xylella fastidiosa is known to transmitted by an insect vector called this glassy winged sharpshooter, Homolodisca coagulata, now name changed to Homolodisca vitripennis. So the answer is Homolodisca vitripennis, because Homolodisca coagulata was not given. Before it is coagulata, now it's called as vitripennis. So the answer is Homolodisca vitripennis. Bacterial food storage organ. All the living organisms generally store their food in some form for utilizing in during off time or whenever we are not getting the food. Sometimes, example, generally, for example, if you take humans have adipose tissue. Whenever we are in fasting, so the energy will be utilized from inside, right? So like that, every bacteria have their own food storage or reserve food storage organ. Generally bacteria can store in the form of poly beta hydroxybutyrate. However, humans have adipose tissue, right? So generally food storage organ of bacteria is poly beta hydroxybutyrate. It is very important. We can also call it as PHB, poly beta hydroxybutyrate. Staining agent of a bacterial food storage organ. So generally bacterial food storage organ can be stained by using an agent called Sudan black B. By using Sudan black BA, we can stain the bacteria. 23. Give an example of Amphitrachus flagella. First of all, we will see what are the different types of flagella in bacteria. On the right side, we can take Atrachus, Monotrachus, Amphitrachus, Peritrachus, right? So Atrachus means no flagella. Example is Xylella. Monotrachus means only one flagella is present, Xanthomonas. Amphitrachus means one flagella to both sides, Pseudomonas species. Peritrachus, flagella all over the body, Irvinia, Pantovia, Escherichia coli, and Clostridium. Lopotrachus, tufts of flagella to both folds. So there are plenty number of flagella at both, for example, like this. Tuft of flagella at both sides. Example, Spirulina. Cephalotrachus, a group of flagella at only one end, Pseudomonas fluorescens. Now what they have asked is the Amphitrachus. Amphitrachus example is Pseudomonas species. 24 is Stewart wilt of corn is caused by. Stewart wilt of corn is known to be caused by Irvinia stewarti. Now we can also call it as Pantovia stewarti. Subpolar flagellum is produced by, we can see here, circumtracheal flagella or subpolar flagella we have seen here, right? So which is produced by the bacterium agrobacterium. Flagella that contains a protein called pyelin. So pyelin is the protein that is present in the flagella. Helical or spiral shaped bacteria is known as spirillum. Generally this spirillum is a helical or spiral shape. Transformation principle was first discovered by transformation principle, conjugation, transformation and transduction. There are a couple of phenomenon were there. I would say recombinations in bacteria, because we cannot say it is a reproduction in bacteria, a recombination in bacteria. Let me see what all the different kinds of recombinations are there. So there are three kinds of recombination: conjugation, transformation and transduction. So conjugation was discovered in the organism E. coli, whereas transformation was discovered in pneumonia, whereas the transduction salmonella. So conjugation was discovered by Lederberg and Tatum in 1946, whereas Griffith discovered this transformation in 28, whereas the transduction was discovered by Lederberg and Zender in 1952. Let's see what is our question. Our question is the transformation principle was discovered by. So just now we have seen the conjugation, transformation and transduction. It was transformation was first discovered by Griffith in 1928. Bacteriocin produced by Bacillus subtilis. So they're asking Bacillus subtilis. There are a couple of bacteriocins are there. So Bacillus subtilis producing a bacteriocin is called subtilicin. What is the staining agent used in electron microscope? In electron microscope, osmic acid can be used generally, maybe other things also used. Generally, among the given options, this osmic acid is used in electron microscope. Counter staining used in gram staining. So for gram staining, safranin can be used as a gram staining, counter stain, right? Endospore staining agent. So what is the endospore staining agent? For food storage organ, for polybeta hydroxybutyrate, we have already seen Sudan black bee. For endospore is a kind of heat resistant spore that is produced by the bacteria, especially gram positive bacteria like Bacillus and Clostridium. So for this identifying this endospore, we can use a staining agent called malachite green. So we'll see the different kinds of staining agents. Endospore staining agent is malachite green, plasmid staining agent is agredendae, capsule staining agent is Indian ink, flagella staining agent is carbol fission, lichen staining agent is arkin, inclusion body staining agent azurea, cytoplasm and nucleus staining agent acetic acid, and bacterial food storage organ is Sudan black bee. We might have saw earlier, right? So the endospore staining agent malachite green. The average time taken for the bacterial binary fission. So binary fission generally take 20 to 30 minutes. Pigment produced by the Erwinia chrysanthemi. So they are asking Erwinia chrysanthemi. Answer is generally indigodine. First of all, we will see what are all the different kinds of pigment that is produced by the bacteria. And these are all the different kinds of pigments that is produced by the plant pathogenic bacteria. So first one is Burkholderia glume produces taxoflavin, Curtobacterium flaccufasciens Pathover poinsettia produces carotenoid, generally yellow and water soluble pigment. Clavibacter michiganensis subspecies michiganensis produces carotenoids. Erwinia herbicola and Erwinia iridovora produces xanthine beta diglucosides. Erwinia chrysanthemi produces indigodine, just now we said earlier, right? And Erwinia rafanseti produces proferazamine. These are very, very important. Erwinia chrysanthemi indigodine, Erwinia rafanseti proferazamine, Erwinia rubifaciens, and Pseudomonas aeruginosa and Pseudomonas fluorescens produces pyoviridin. Xanthomonas produces xanthomonidin, Pseudomonas aeruginosa produces pyonin, and Pseudomonas apiaceae produces phenazine, and Pseudomonas aeruginosa produces pyorubin and pyomeladin. These are all the different kinds of pigment that is produced by the plant pathogenic bacteria. Next one is the test used for the identification of Pseudomonas. Generally, for this identification of Pseudomonas fluorescens, there is a specialized test called LOPAT test. I think there is an expansion for this LOPAT, like actually I forgot, but there is an expansion for every single word, LOPAT, LOPAT. But generally this LOPAT technique is used for the identification of Pseudomonas fluorescens. So the leaf witness period of apple scab. So generally the leaf witness period of apple scab is 9 hours. Why 9 hours? Like generally for identification I'm telling, apple is a 5 letters, scab is 4 letters, 5 plus 4 9 letters, so it is a 9 hours, like that. But it also varies according to the temperature and humidity and other kinds of condition. So based on the temperature, the leaf fitness period also varies, but generally the standard leaf fitness period of apple scab is 9 hours. So what is the molecular weight of amylovorin? So amylovorin has a molecular weight of 165,000 daltons. I'm sorry. Fire blight of apple and pear pathogen requires which acid for the growth factor? This is also very important. Nicotinic acid. So for the presence of nicotinic acid is very important for the growth and germination of this fire blight of apple, which is caused by Irvinia amylovora, right? Which of the following element has a highest composition in a microbial cell? In a microbial cell, what is the general highest composition? Carbon, nitrogen, hydrogen, oxygen. Generally, it contains more amount of carbon. Name the pathogen that enters into the trichomes. So on the next slide I will tell you the modes of entry of different plant pathogen. That is a very important. Every year we must expect at least one question from that. Generally the answer is bacterial canker of tomato can enter into the plant cells through trichomes. And on the later slide, as I told, there are different methods of entry of plant pathogenic bacteria were there. Berg's manual of systemic bacteriology they are asking. So if it is a systemic bacteriology, make sure the answer is 1984. If it is a determinative bacteriology, the answer is 1923. So D comes first, S later. You can remember in that way. 1923 determinative bacteriology, systemic bacteriology 1984. We can remember in that way. And these are all some mode of entry of plant pathogenic bacteria. So through hydrothodes, black rat of cabbage and bacterial leaf blight of rice gains entry. On the right side I have given causal organisms, you guys might have known. Floral parts, fire blight of apple and pear. Through lentils, potato common scab and as well as soft rot of vegetables. Through trichomes, bacterial canker of tomato, Clavibacter michiganensis subspecies michiganensis, just now we saw earlier, right? And through stomata, bacterial leaf streaks, citrus canker, angular leaf spot of cotton, also known as black arm of cotton, also known as bacterial blight of cotton, and common blight of beans and as well as leaf spot of tomato. These are all gains entry through stomata. And through wounds, soft rot of vegetables, stalk rot of maize, crown gall of fruits, stone fruits, and penicillium species. These are all known to enter through wounds. And through insect vectors, because we might have studied in plant virology, majority of the plant pathogenic bacteria enters through insect vectors. But there are a couple of bacteria also there, they enters through insect vector. I think we might have studied earlier, like this is Pierce disease of grapevine, Xylella fastidiosa. It is known, sorry, not entry, transmits, I would say like transmission. So Xylella fastidiosa, that is Pierce disease of grapevine, glassy winged sharpshooter, Homolodesca coagulata, we might have studied earlier, right? So now through insects, bacterial wilt of corn, which is known to be caused by Pantovius stewardii, known to be transmitted by corn flea beetle, Chitocnema pulcharia, bacterial wilt of cucumber, Irvinia trachyphylla, and through root hairs, root nodule bacterium can enter entry, and through nematodes like wheat ear cockle, that is Rathibacteria tritici, generally associated with a nematode called Anguna tritici, and bacterial wilt of carnation, that is Paraborcolderia caryophyllae. These are actually enters through nematodes. Next one, bacterial blight of rice is an epidemic in Bihar in the year. It is very severe in Bihar during the year of 1963. It is a very important question. And constant is actually transmitted by. So constant is transmitted by an insect vector called Dalbulus maedis. G plus C content of Irvinia, which means guanine and the cytosine content of Irvinia. So the GC content of Irvinia ranges from 50 to 58%. IMTech, Institute of Microbial and Technology, is actually located. It is a very famous institute. Generally serves cultures and maintains various kinds of culture, and one of the largest and biggest microbiological center where many cultures are located. And it is actually located in Chandigarh, India. Azotobacter was discovered by Bajorniki. That's why if you see in the history, this question was there, like Azotobacter was discovered by Bajornik, and there are a couple of other species also discovered by him. Effective antibiotic against rice blast. Rice blast is a fungal disease, but generally the Fungicide that is recommended for rice blast is a tricyclazole, selling outside with the trade name of blastin or blastin. Yes, so various kinds of trade names are there, but generally in another antibiotic can be used for this rice blast, that is agromycin or agromycin 100. The concentration of antibiotics used in plant disease management. So for antibiotics it is used in ppm, which means 0.01% concentration. 100 ppm or in between 100 to 200 ppm. Fluorescent pigment produced by Pseudomonas glume, they are asking. So they produce pigments called taxoflovin and fervanulin. How many rings are present in gram-positive bacteria? So gram-positive bacteria contains 2 rings, whereas gram-negative bacteria contains 4 rings. Bacteria having a peritracheous flagella, asking example, right? So we know very well like Irvinia. So this is the best example of bacteria which consists of flagella all over the body, that is called peritracheous flagella, Irvinia, right? Examples of star-shaped bacteria. Star-shaped bacteria is known as the Ancylomicrobium. Irvinia belonging to the family. Irvinia generally belonging to family Enterobacteriaceae. So gram staining method that was given by Christian Gram in the year, important one, it was given by Christian Gram in the year of 1884. The major difference between phytoplasma and spiroplasma. So the primary difference between phytoplasma and spiroplasma, they are different in shape, they are different in cultivability, and they are different in motility. So the answer is all the above. Most of the bacteria are rod-shaped except some species. For example, Streptomyces is not rod-shaped, right? So bacterial cell wall is made up of bacterial cell wall is generally composed of NAG and NAM. We might have studied earlier, right? N-acetyl glucosamine and N-acetyl muramic acid constitute to form beta 1,4 linkage, right? Test used for the identification of a bacterial disease. For all bacterial disease, we used a test called Wuzo test. So if the Wuz is present, it is a bacterial disease. Penicillium notatum is now called name changed. For example, if you go internet and just type Penicillium notatum, you will get Penicillium chrysogenum. Now it is name changed to Penicillium chrysogenum, and moreover nowadays what we are using penicillin is actually now they are extracting from Penicillium chrysogenum because it gives 100 times more yield than the Penicillium notatum. So now it's called as Penicillium chrysogenum. Endotoxins are found in generally gram-negative bacteria, gram-positive bacteria in exotoxins. Endospore forming a bacterial gram-positive bacteria, they are asking. Agrobacterium, Clostridium, Ralstonia, Serratia, all are gram-negative except this Clostridium. Clostridium and Bacillus, they produce a heat-resistant spores are known as endospores. So the process of elimination of plasmid is known as curing. Curing is a process where plasmid can be eliminated. Bacterial blight of cotton is external, internal, A and B, none of the above. Generally, this bacterial blight of cotton or black arm of cotton is known to be both the external and internal seed-borne pathogen. Bacterial blight of cotton, same both internally and externally seed-borne pathogen. The first broad spectrum antibiotic was discovered. Broad spectrum, they are asking. The first antibiotic was discovered by penicillin only. Since they have asked first broad spectrum antibiotic, it is streptomycin. Mode of action of TAP toxin. It actually inhibits glutamine synthase. Maximum value count of bacteria can be seen in, I repeat, maximum value count, in which phase the maximum amount of viable bacterial cells will be present. Generally in log phase, more amount of viable cells will be present. Genes responsible for pathogenicity and the elicitation of hypersensitive response or hypersensitive reaction are known as, generally HRP genes are responsible for this hypersensitive response and all the things that is related. HRP genes, okay. Actinomycetes generally produces exospores among the given options, okay. We must answer according to the options given, right? So in which phase of bacterial growth time taken for doubling the number of cell is constant for a period of time? In which phase where the doubling time, time taken for doubling the number of cells is actually constant for some period of time, they are telling that it is a log phase actually, because it need to adapt to the environment. So it will take a little bit to adapt, and so that they started doubling. Number of chromosomes in a bacterial cell. Bacterial cell contains only one chromosome. A bacterial cell exists aneuploid, tetraploid, haploid and diploid. I think it's haploid. Exospores are formed by which of the following bacterial genus? They are asking Streptomyces, Bacillus, Clostridium and Actinomyces. Delovibrio parasitizes. Delovibrio generally parasitizes a gram-negative bacteria. So if you see the definition also, a gram-negative bacteria that parasitizes another gram-negative bacteria is known as Delovibrio. So it was first discovered by Slope in Slope in 1962 probably. The genus Xanthomonas belonging to the class. It is belonging to the class Gamma proteobacteria. And Mucovilta of banana is caused by, it is caused by Ralston genus. F plasmid is transmitted from one bacterial cell to another bacteria. Another bacterial cell is through conjugation only, because where both cells are contact with the help of conjugation pilus, where the plasmid moves from one bacterial cell to another bacterial cell. So the answer is conjugation. Lethal yellowing of coconut. Coconut lethal yellowing is known to be caused by phytoplasma. It is a phytoplasmal disease and known to be transmitted by leaf of Permentus protus. Hairy root of apple is caused by, it is actually caused by agrobacterium. Crown gall of apple and hairy root of apple both are caused by agrobacterium. Which of the following genera causes most of the wilt diseases in vegetables? They are asking vegetables. Generally, Ralstonia causes a majority of the wilts in vegetables. The wildfire toxin is secreted by, it is secreted by a bacteria called Pseudomonas syringae pathovarictivaci. That's why it's called tab toxin or wildfire toxin. Who is regarded as the father of plant bacteriology in India? They are asking. In India, M.K. Patel is generally considered as the father of plant bacteriology. K.C. Mehta, father of Indian Trust. E.J. Butler, father of Indian plant pathology. B.V. Munkur, who established Indian Pathological Society. Right. So the name of Xanthomonas was coined by, this is also very important. There are a couple of things like who coined and all those things will be there. So the name Xanthomonas was actually coined by Dawson probably in 1920, I think so. 1920, maybe 1919 or 17, I think so. Center rot of onion is caused by, it is caused by Pantovius ananatis. Bacterial wilt of dry beans is caused by, it is known to be caused by Cortobacterium flaccum pathovar flaccum and it is survive around 24 years in laboratory conditions, it is mentioned. Black chaff of cereals is caused by, it is caused by Xanthomonas campestris pathovar andalusa. Fluctuation test was actually developed by, this fluctuation test was developed by Lurie and Delbruck for the identification of mutations in bacteria, and also later Delbruck alone discovered these mutations in virus as well. Fire blight of apple pathogen can enter into the plant through nectaris. Casugamycin is an antibiotic isolated from a bacteria called Streptomyces casuganensis. Leaf gall of tobacco is caused by Rhodococcus fascinans. What are the techniques used for isolation and purification of bacteria? Pore plate, strict plate, spread plate. Okay, these all are used for the isolation and purification of plant pathogenic bacteria. Sugarcane ratoon stunting disease is transmitted. It is very important. Sugarcane ratoon stunting is caused by Lifesonia xylem, subspecies xylem. Before it is called as Clavibacter xylem, subspecies xylem, and it is known to be transmitted by infected seeds. Angular leaf spot of cotton is otherwise called as black arm, bold rot, black rot, all the above. So the answer is all the above, because it is also known as black arm, also known as bold rot, also known as black rot, all the above. Dickia dadanthi causes the disease name. So Dickia dadanthi causes which disease? Substrate of potato. A virus that affect bacteria are called as, I repeat, a virus that affect bacteria is called as bacteriophages, or I would say a virus that infect bacteria is known as bacteriophages. The term bacteriophage was given by Harely. The meaning of bacteriophage is bacteriophage. Phase means eater, bacterial eater. A virus that infect bacteria is known as bacteriophage, right? Soft rot disease of fruits and vegetables is caused by, it is known to be caused by Yersinia. Which one is called as a wonder drug? It is penicillin. So in 1928 it was discovered entire British army during the World War II, 1940s. It was served with penicillin. So to some kind of infection and all those things, it was miraculously cured this. So that's why it is called as a wonder drug. First virulence gene was isolated from. They're asking isolated from where? Actually, the first virulent gene was isolated from bacteria. Later, fungal virulent gene in 1991 by Dewitt, but bacterial virulent gene was isolated by Staskowitz probably in 1984 itself. Fungal virulent gene is 1991, right? Ice nucleation active bacteria was discovered by. It was first discovered by Mackey et al. The first antibiotic penicillin was discovered in the year. It is discovered in the year 1928. The role of enzymes in plant pathogens was reported by. So the role of enzymes was reported by Boran, I think. Avirulence gene for Pseudomonas syringae, pathovar glycinia DC300 was first isolated by. It was first isolated by B.J. Staskowitz. That is, as I told you, Pseudomonas syringae, pathovar glycinia, 1984, I think. Who received a Nobel Prize for mass multiplication and bringing into the chemotherapeutic use of penicillin. Flore and Herbert B. Chain, Ernest B. Chain. I think we have discussed in the history itself. So for in 1945 in the physiology and medicine, 3 people got Nobel Prize. One is Alexander Fleming for the discovery of penicillin, and the other 2 scientists is Flore and Ernest B. Chain. These 2 got just because of a mass multiplication process. Okay. Bacterial chromosome is called as genopore. That was given by Riss. Hans Riss in 1961. He called bacterial chromosome as genopore. Bacteriophages of Bacillus keratovirus was isolated by Cones and Cotilla. Pathovar system of taxonomy of bacterial plant pathogenic bacteria was first used by Dye et al. So this pathovar system actually introduced by Dye et al. In 1980, I think. Next one, who received Nobel Prizes for the studies of bacteriophages and established the modern molecular biology? Probably Delbruck, Herschel, Lurie. I think everyone got Nobel Prize. So probably the answer is all the above. I think so. Conjugation gene transfer in bacteria was actually demonstrated by Chatterjee and Starr, who actually demonstrated this conjugated gene transfer. The 8th edition of Berg's Manual of Determinatory Bacteriology was edited by Buchan and Gibbonson. Bacterial filters was invented by, so it was actually invented by Charles Chamberlain. That's why it is called the Charles Chamberlain filter. A group of cocci which was arranged in an irregular shape is known as Staphylococcus. Budding bacterial genus. Among the given options, the budding bacterial genus is Rhodomicrobium. The number of cryptic plasmids are present in Irvinia stevati. This was a little bit, important one. Generally, bacteria contains 1 plasmid, but there is a bacteria called Irvinia stevati, which consists of 11 to 13 cryptic plasmids. The race 1 of Ralston is Solanacearum. In fact, we might have discussed this, right? Race 1 Solanaceae, race 2 Musaceae or banana, race 3 potato, race 4 ginger, race 5 mulberry. Since they have asked race 1, it is a Solanaceae. Olive quick decline is caused by bacteria generally. Oleander leaf scorch is caused by it is a Xylella fastidiosa. We know we have already discussed this, like Pierce disease of grapevine, citrus variegated sclerosis, oleander leaf scorch, alfalfa dwarf, almond, phony peach leaf curl, all are caused by the Xylella fastidiosa, and it is also known to be the first plant pathogenic fungus to become bacteria to be completely genome sequenced. Pierce disease of grapevine is also caused by Xylella fastidiosa. The next question is, Pierce disease of grapevine is transmitted by. It is known to be transmitted by glassy winged sharpshooter. The scientific name is Homolodisca coagulata, and now it is called as Homolodisca vitripinis. Stewart wilt of corn is caused by. Irvinia stewati. Now it is called as Pantovia stewati, both A and B. Just now discussed Stewart wilt of corn is caused by, right? Irvinia stewati or Pantovia stewati. 6-pile is produced by. It is actually produced by male bacteria. Iron producing bacterial genus Gallionella. Bacteriosis was actually discovered by Andrew Gracia in 1925. Bacterial genus which causes a maximum number of plant diseases. It is very, very important. Generally, the Pseudomonas causes more number of plant diseases rather than Xanthomonas. Decolorizing agent used in a gram staining of bacteria. So 95% ethyl alcohol can be used. Example of xylem inhibiting gram positive bacteria they are asking. So it is sugarcane ratoon stunting. This is actually FVB, fastidious vascular bacteria. Two types will be there: xylem inhibiting and phloem inhibiting. In xylem inhibiting, two types will be there: gram positive and gram negative. So gram positive example is probably sugarcane ratoon stunting. Gram negative xylem inhibiting is Pierce disease of grapevine. When it comes to phloem limited, only one type is there, that is a gram negative citrus uh, candidatus liveribacter asiaticus. Citrus greening, I think citrus greening comes under phloem limited gram negative bacteria. 124. Why wall-less bacteria is named as L form? So why it is named as L form? Because discovered by Lister to honor the Lister Institute. L stands for less. So all the above. So the answer is generally to honor the Lister Institute. Where Kleineberger Nobel was working. Actually, this L form of bacteria was discovered by Kleineberger Nobel. To honor the institute where she was working, so this named as L form of bacteria, which means Lister Institute. So L form of bacteria was discovered by, just now I told you, it was discovered by M.E. Kleineberger Nobel. Fluctuation test is used to discover. Generally, this fluctuation test is recommended for the identification of bacterial mutations. So bacterial mutations were first identified Luria and Delbruck. Okay. Next one is who are considered as the founding father of phased genetics. So both Luria and Delbruck, because in history we might have studied this Luria and Delbruck, they discovered bacterial mutation. Later Delbruck alone discovered the viral mutations in 1939 and various kinds of things we might have discussed in history, right? So who is considered as founding fathers means Luria and Delbruck. So the answer is both A and B. Pigment produced by Irvinia refonceti. So we have already discussed it. Still I will tell you, proferazamine. Proferazamine is the pigment that is produced by this Irvinia refonceti. And these are all the various pigments we might have already discussed: Burkholder, glumate, taxoflavin, Curtobacterium flaccum fasciens, carotenoid, Clavibacter michiganensis subspecies michiganensis carotenoid, Irvinia herbicola, zeaxanthin, Irvinia chrysanthemum indigoda, Irvinia refonceti, proferazamine, Irvinia rubrifacin, Irvinia pseudomonas aeruginosa, pyoverdin, xanthomonas, Irvinia aeruginosa, pyocyanin, Pseudomonas epiphenoxylin, Pseudomonas aeruginosa, pyorubrin, and pyomelanin. These are all the various kinds of pigment that is produced by the bacteria. And the next one is G plus C content of streptomyces. So the G plus C content of streptomyces is probably in between 60 to 70%. So the next one is xylem inhibiting a plant pathogen. Which of the following is a xylem inhibiting? Xylella fastidiosa, Pseudomonas cerevisiae are among the given options. This is Xylella fastidiosa is the best example for xylem inhibiting gram negative bacteria. Okay. Black rot of cabbage can enter into the plant cells through. We have already discussed the different bacterial plant pathogen that gains entry through. Generally, this bacterial black rot of cabbage, which is caused by Xanthomonas campestris, Pathover campestris, gains entry through hydrothodes. Name a plant pathogen that enters into the plant cells through lenticels. So potato common scab. Fire blight of apple can generally spread through insects, ants, pneumocoria, all the above generally. Pathogen responsible for a 100-fold increase in oxidants of potato. So there is a pathogen called Pseudomonas solanacearum. If it is get infected, the amount of oxidants content which actually increases 100-fold increase in the potatoes. So this is Pseudomonas solanacearum. So the first antibiotic to be purified in a pure form by glyotoxin. Endospores are highly resistant to heat due to the presence of an acid called DPA, dipicolinic acid. If you see the endospore, bacterial endospores, bacterial endospores are generally produced by the pathogens like Bacillus and Clostridium. So this Bacillus and Clostridium, they will produce an acid called dipicolinic acid that helps, which contains more amount of calcium ions. So that will be helpful for withstanding against high temperatures. So the acid is dipicolinic acid. Endospores are individually discovered by corn, Robert Koch, John Tyndall. Actually, endospores are individually discovered, all the above. Kohn in 76, Robert Koch in 77, John Tyndall in 1978. I think all are. Oh no, John Tyndall in 76, Robert Koch in 77, Kohn in 1878. They all are discovered this endospores individually. So all the above answer is. Acinides are produced by general acinides are produced by cyanobacteria. The bacteria responsible for blue color production of milk. This is also very important. Couple of the bacteria produce blue color, couple of the bacteria produces green color, couple of the bacteria produces red color, green color, various kinds of things will be there. I have given here. So blue color pigment is actually produced by Pseudomonas syncytia. I repeat, blue color pigment. When it comes to red color pigment is produced by Serratia marcescens, and whereas green color is produced by Pseudomonas aeruginosa. This is very very important. Blue color means Pseudomonas syncytia, red color means Serratia marcescens, green color means Pseudomonas aeruginosa. And next one is sugarcane ratoon stunting is caused by. So sugarcane ratoon stunting is caused by Lifesonia xylem subspecies xylem. The previous name of Lifesonia xylem subspecies xylem is the Clavibacter xylem subspecies xylem. So if you just go and type in the internet, you will find both this Clavibacter xylem subspecies xylem, now renamed as Lifesonia xylem subspecies xylem. In 2021, this question was asked. Sugarcane ratoon stunting is previously caused by or previously called as Clavibacter xylem subspecies xylem. So the answer is both A and B. And which bacteria convert ammonical form of nitrogen to nitrates? They are asking. Nitrosomonas. We might have studied in 12th itself, right? So locomotion type of non-flagellated bacteria. So if generally bacteria having a flagella for the locomotion, what if the flagella is not possessed by the bacteria? So they can locomote by using a mechanism called gliding motility. Through the gliding motility, they can locomote. Bacterial wilt of cucumber is caused by Irvinia trachyphylla. Citrus greening is transmitted by psyllids, Asian psyllids, Diaphorina citri, I think so. And what is the first plant bacterial disease to be reported in India? It was the first plant bacterial disease, bangle blight of potato. So the bangle blight of potato that was reported by E.J. Butler. This is the first known reported plant bacterial disease in India. Sesamum phyllodi is transmitted by. It is known to be transmitted by leafhopper. And enzyme required for the photoreactivation of nucleic acid. So there is an enzyme called phytolase which is actually responsible for the photoreactivation of nucleic acid. Agrobacterium is actually belonging to the family. This agrobacterium, well known, it is belonging to the family Rhizobiaceae. That's why now it is named as Rhizobium radiobacter, right? And before conjugation, the condition of plasmid DNA. So before conjugation, the condition of plasmid DNA is a double stranded. When later they become single stranded, again they will become double stranded. That is secondary. But the before conjugation, the state of the plasmid is double stranded. So that's why in definition we might study like this. Plasmid is a circular double stranded extra chromosomal DNA that is present in the bacteria. Circular double stranded extra chromosomal, right? So the ratio of RNA and DNA in a gram positive bacteria, generally in gram positive bacteria the ratio is about 8:1, 8 parts of RNA and 1 part of DNA. Bacterial blight of rice is caused by Xanthomonas oryzae, Pathovar oryzae. Bacterial leaf streak means Xanthomonas oryzae, Pathovar oryzicola. Phytoplasmal disease are generally transmitted by, majority of the phytoplasmal diseases are known to be transmitted by an insect vector called leafhopper. Which of the following organism is considered as the natural genetic engineer? So we know very well agrobacterium. Generally this agrobacterium is considered as a natural vector, right? Since we have given agrobacterium radiobacter and agrobacterium tumefaciens, so the answer is both B and C. Most of the plant pathogenic bacteria are rod shaped, right? So crescent phase can be observed, and this is one of the important phase when we see the bacterial leaf blight of rice, which is caused by Xanthomonas oryzae, Pathovar oryzae. There are 3 stages are there: yellow leaf phase, crescent phase, and wilting phase. This crescent phase also known as wilting phase, I think so. So this crescent phase is the most important and deadliest
phase when it comes to the rice bacterial disease. So this crescent phase can be observed in rice bacterial leaf blight, which is caused by Xanthomonas varaceae, pathovar varaceae. Fasciole toxin is produced by Pseudomonas syringae, which is produced by Pseudomonas syringae. The next one is angular leaf spot of cotton is caused by Xanthomonas campestris, Pathovar campestris, Pathovar malvacearum generally. Xanthomonas campestris, Pathovar malvacearum. Sorry, actually Xanthomonas campestris, Pathovar campestris means that goes black rat of cabbage. How the black rat of cabbage can enter into the plant cells through hide thoughts, right? So just correlate the questions so it will be easy. The next one is the shape of growth curve of bacteria. Generally linear sigmoid bimodal saturation curve. Generally the growth curve shape is bimodal in shape. The next question is Irvinian Trachyphylla, bacterial wilt of cucurbits pathogen, generally called bacterial wilt of cucurbits, which enters cucurbits pathogen is spread by, how it is spreading? Generally it is known to be spread by insect vector like cucumber bark beetle, Chitonema pulcheria, other thing. No. Insects like Diabrotica duodecim Diabrotica victata insect vector is there, right? And which of the following organism that parasitizes gram negative bacteria? A bacteria which parasitizes which parasitizes another gram negative bacteria is known as a Dello vibrio. So Dello vibrio was first time discovered by Slope 1962. HFR cell. What is HFR? High frequency recombination cells, right? So HFR cells are high frequency recombination cells are produced as a result of conjugation, transformation, transduction and transfection. So in conjugation mechanism only where two bacterial cells are come in contact with each other with the help of conjugation pilus or sex pilus, right? So generally these HFR cells are produced as a result of conjugation. So hairy root of apple is caused by agrobacterium. There are two strains of agrobacterium we know very well, Agrobacterium tumefaciens and Agrobacterium rhizogen, Agrobacterium radiobacter, right? Sorry, Agrobacterium tumefaciens and Agrobacterium radiobacter. This radiobacter is generally used as a biocontrol agent. This tumefaciens is a plant pathogen. Of course, other species also there in agrobacterium. Generally, this hairy root of apple is also caused by the species of agrobacterium. Then nutritionally, the plant pathogenic bacteria in general, what the plant pathogenic bacteria are, like photoautotrophs, photoheterotrophs, or chemoautotrophs, or chemoheterotrophs. Generally, in general, majority of the bacteria are photoheterotrophs in general. So the source of polybeta hydroxybutyrate. First of all, what is this polybeta hydroxybutyrate? Is a bacterial food storage organ is known as polybeta hydroxybutyrate, also known as beta hydroxybutyrate. It contains a little or more amount of carbon. Antonio Luinac was the first person to see the bacteria in the samples of in which sample he had saw the this bacteria. So generally he saw in dental plaque. Give an example of xylem limited or xylem inhabiting a gram positive bacteria they are asking here. In xylem two types are there, xylem positive, xylem negative. In phloem again most important one type, that is xylem phloem gram negative. So in xylem gram positive are there and gram negative are there. See, xylem gram positive means for example, we can take the examples of Livesonia xyli subspecies xyli, which causes sugarcane rottum stunting. Xylem gram positive. Xylem gram negative means a pierced disease of grapevine or pierced disease of grapevine, which is caused by Xylella fastidiosa. Phloem gram negative means Candidatus liveribacteria asiaticus, which causes citrus greening. Here they have asked xylem gram positive, which means Livesonia xyli subspecies xyli. So this Xylella fastidiosa is a xylem limited gram negative, okay. And this Candidatus liveribacteria asiaticus, which causes citrus greening disease, is an example of phloem limited gram negative bacteria. Okay. The next one is the citrus variegated chlorosis is caused by Xylella. Not only this pierced disease of grapevine, there are a couple of list of important diseases that all are known to be caused by this Xylella fastidiosa. I will tell you. The first one is pierced disease of grapevine, citrus variegated chlorosis, almond leaf scorch, alfalfa dwarf, phony peach disease. These are all are the and couple of other diseases also there. These are all of the list of diseases that is known to be caused by Xylella species. Generally, this pierced disease of grapevine is known to be transmitted by an insect vector called glassy winged sharpshooter. Homolodisca coagulata, now name changed to Homolodisca vitrepinis, right? We might have studied somewhere, right? So generally this citrus variegated chloros is also caused by Xylella species. And the selective medium recommended for Pseudomonas. For Pseudomonas, Kingsby medium is recommended. It is the most suitable medium, right? So the pigment produced by the Pseudomonas fluorescens. What is the kind of pigment that is produced by? They produce a pigment called pyoviridin. That's why they produce a kind of pigment. Okay. Xanthomonas varese, Pathover varese was completely genome sequenced by. So let me tell you the first plant pathogenic bacteria to be completely genome sequenced by this Pierce disease of grapevine or Pierce disease of grapevine, Xylella fastidiosa. In later years, other bacteria like rice blast and other leaf streak also have been genome sequenced. But this Xanthomonas varese, Pathover varese, which causes rice bacterial leaf blight, was the first time genome sequenced by B.M. Lee in 2005. It's a very important one. Xanthomonas varese again same BM Lee. Next one Xanthomonas campestris, Pathover campestris, that was sequenced by. So which causes black rot of cabbage, which was sequenced by Anna da Silva in 2002. Big bud of tomato is caused by tomato big bud is a phytoplasmal disease generally. Size of TA plasmid majority mostly in common. The size of plasmid, the most common size of plasmid is 200 kb. Rice bacterial leaf blight, which causes Xanthomonas varese, Pathover varese, is epidemic in the state. In which state it is epidemic? It was epidemic in the state probably Punjab 1980, I think so. And later in Tamil Nadu around 1985-1990s time, this rice bacterial leaf blight is again epidemic, but first in Punjab I think so. Name the first plant pathogenic bacteria whose genome is completely sequenced. Just now we have discussed the first plant pathogenic bacteria that is completely genome sequenced is Xylella fastidiosa. Who coined the term murin? So we might have studied this murin. What is murin? Bacterial cell wall is composed of peptidoglycan, or we can also call it as mucopeptide or murin, right? So who coined this murin? Generally this murin was coined by Vidal et al. 1960, I think so. Lac-Operon concept of gene regulation was given by, so it was given by Jacob and Monod. What is the world's first living organism to be patented? The world's first living organism to be patented is Pseudomonas putida. We can also call it as giant Pseudomonas. The Pseudomonas putida, we can also call it as a superbug, was designed for, it is actually designed for oil degradation. For example, if there is some oil spillage or during the transportation of oil through the seaport, so sometimes there might be accidents, right? So in such cases, all the oil that was there in the ship got spilled out. So to clean this oil spillage or oil degradation, Chakrabarti et al. They used a bacteria called Pseudomonas putida. It will be very helpful to degrade. Okay. In India, next question. In India, plant bacterial research was initiated by. In India, it was first initiated by M.K. Patel only. That's why we called M.K. Patel as father of Indian plant bacteriology, right? Bacterial blight of rice was first reported in which state of India? In Uttar Pradesh, this bacterial blight of rice was first reported. Which of the following scientists stated that microorganisms are pathogenic if they are toxigenic? I repeat once again. Who gave this terminal, this statement? Microorganisms are pathogenic if they are toxigenic, which means if they are producing toxics, automatically they are definitely pathogenic. Such a statement was first time given by government. Which of the following plant pathogen cannot be cultured in the laboratory condition? Best example is Candidatus phytoplasma. That's why it is called Candidatus. Candidatus is the state where it is designed to something that is cannot be cultured. So this Candidatus phytoplasma cannot be cultured in the laboratory. Whereas Spiroplasma can produce poached egg or fried egg kind of colors, right? So which of the following bacteriophage is a lipid envelope? Which bacteriophage? There is a phase PM2. This bacteriophage contains a lipid envelope. Which of the following bacteriophage is a lipid? Just now we discussed phase PM2, right? And next one is the sequence of lytic cycle of bacterial phase. General sequence is absorption, penetration, replication, maturation, and liberation. This is the general sequence. ATP value of bacteria. The bacterial ATP value is, I think, around 10. So Dello Vibrio is around 15 to 20, I think so. Just a minute. ATP value of bacteria is 10 probably, and Dello Vibrio is I'm little bit doubtful, probably 20 to 30. I'm not sure, probably. Next one is the signal mechanism which controls the physiological functions in response to the population size. I repeat, signal mechanism which controls the physiological functions in response to the population size is known as quorum sensing. Fatty acid profiling depends on separation of fatty acids that is actually based on size and conformation. Gram negative bacteria that are actually comes under which division? Gram negative bacteria comes under grass liquids. Gram positive bacteria that comes under gram positive bacteria comes under firmicutes. Mode of action of TAB toxin. So it inhibits actually glutamine synthesis. It is a very important. The mode of action of TAB toxin is it inhibits glutamine synthesis. Number of polar flagella for Dello vibrio. So the Dello vibrio contains one polar flagella. Botulism is a toxin that is a toxin is a generally what it is, protein or lipid or sugar or vitamin. It is actually a protein which causes botulism. Okay, who named the bacillus? The name bacillus was the first time given by Ehrenberg. The term vaccine was coined by Edward Jenner, I think so. That's why it's called is considered the father of enzymatology or something else. Uh, Light yellowish color seen in the cow milk is due to the presence of a pigment called keratin. Same just now we discussed. The light emitted by luminescent bacteria is mediated by the enzyme. Which enzyme is responsible for this light emission? I think lactose dehydrogenase. I think so. Mycobacterium tuberculosis was first time discovered by. It was first time discovered by Robert Koch, and it is also one of the example for both gram positive and gram negative bacteria. The enzyme responsible for decomposition, proteolytic enzymes. Drug resistance in bacteria is mainly determined by the which factor actually determines. So it is actually probably determined by lysogenic factors, I think so. Bacterial wilt of cucumber is caused by, it is known to be caused by Pseudomonas syringae pathover lacrimens, very important one. The rice gene which confers the resistance against Xanthomonas virus, a pathover virus that codes LLR protein kinases. So this is Xanthomonas 21. It's a very important one. Which bacterium produces yellow color colony? So yellow color colonies are produced by which of the following bacteria? It is produced by Xanthomonas. Xanthomonas produces yellow color. And who proposed the first genus based on the physiological characters? For the first time, this Weijernick actually started classifying this based on this physiological characters. So Mycobacterium leprae was discovered by Hansen. Meaning of amylovorus. Generally, the meaning of amylovorus is a starch eater. Amylovorus. Amylo means starch. Vorus means eater. Starch eater. Most of the insect vector transmitting bacterial disease is having a which type of mouthparts? They are having a sucking and biting type of mouthparts. Pasteurization is also known as high temperature short time method because at high temperature of 62.8 degrees Celsius for 10-15 minutes, right, we are doing it, right? So that's why it is called as a high temperature short term method. Okay, wildfire of tobacco producing toxin, the wildfire of producing a toxin called TAB toxin, T toxin, we can also call it, right? The Streptococcus pneumonia was actually isolated from. It is isolated from. Isolated by. Sorry, isolated from. It is isolated by Louis Pasteur. Halo blight of beans is caused by. It is caused by Pseudomonas syringae pathover fasciolicola. The generation time is what? What is the generation time? The time required for the cells to divide, the total division of cells during the lifetime, total number of cells formed, number of none of the above. The generation time is means total number of cells is formed is known as generally generation time. Marginal chlorosis of strawberry is caused by, it is caused by Candidatus flumibacter. Short rot of corn is caused by, it is caused by Dickia zeae. Anthracis, Bacillus anthracis was isolated by, it was first time isolated by Robert Koch only. Okay, Rickettsia-like organism was first discovered by Rickettsia-like organism was first discovered by Ricketts. That's what it's called as a Rickettsia-like organism. The most active stage of sigmoid curve of bacteria in which maximum growth is attained. They're asking sigmoid curve of bacteria in which a maximum growth is attained. There is a stage called probably this log phase where the maximum like lag, log, stationary, and decline phase, right? So in the log phase, there will be a maximum growth of this bacteria. Penicillin is a, what is penicillin? Normally we know it is a antibiotics. The definition of antibiotics, there are secondary metabolites compounds are produced by bacteria or maybe fungi, right? So doubling time of rickettsia by binary fission. Generally, bacteria took 20 to 30 minutes for binary fission, but this rickettsia-like organisms, they took at least 8 hours to reproduce. I mean, become half. Pseudomonas aeruginosa was first named by Pseudomonas aeruginosa first named by Schroeder and Gesad. Example of alpha proteobacter. So Rhizobium, Azotobacter, Glauconobacter, all are the examples of alpha proteobacter. And the next one is examples of beta proteobacter. Acidovox, Burkholderia, Ralstonia, all are the examples of beta proteobacter. Examples of gamma proteobacter, again same, Erwinia, Pectobacterium, Pseudomonas, all are the examples of gamma proteobacteria. What is the type of ribosomes present in the bacteria? The type of ribosomes present in the bacteria is generally 70th type of ribosomes are present. Growth is a closed system affected by the nutrient limitation, and the waste product accumulation is generally called as a batch culturing. 230. The bacterial cell with a particularly damaged cell wall is known as spiroplast, gram-negative bacteria having, gram-positive bacteria having protoplast, right? Partially digested cell wall something. Completely digested cell wall something, right? The best medium for the production of penicillin. So steep liquor, I think so. I'm not sure. Steep liquor, I think so. Maybe. Xylem inhabiting gram-negative fastidious vascular bacteria. We have already discussed. Gram-positive means that is what we have discussed, that Shorkey and Raton standing. Gram-negative, since they have asked Pierce disease of grape vine, Pierce disease of grape vine, right? Phony peach leaf, almond leaves. Yeah, all are caused by Xylella fastidiosa only. So the answer is all the above. Because this phony peach leaf curl or Pierce disease of grape vine, almond leaves, all are known to be caused by Xylella fastidiosa only, which is the best example for gram-negative xylem limited bacteria. Serratia marcens actually comes under phloem inhabiting gram-negative bacteria. Industrially important antibiotics producing organisms shall be isolated by which technique? They are isolated by a technique called crowded plate method. So the scientific name of spotted cucumber beetle, Diabrotica duodecimpunctata, or we can also call Diabrotica vittata, Acailama vittata, that is another thing, right? So since they have asked spotted cucumber beetle, the scientific name is Diabrotica duodecimpunctata. Example of soil inhabiting bacteria. So Agrobacterium, Streptomyces, Ralstonia, all are soil inhabiting bacteria only, right? So all the above, right? Example of soil invading bacteria. Again, this is Xanthomonas, Erwinia, and Clavibacter. All are the examples of soil invading bacteria. So the answer is all the above. So best examples of traditional microbial fermentation product is. So fermentation product, they are asking among the given option, this vinegar is the best example for microbial fermentation product, right? Of course, other things also, but generally this vinegar is the microbial fermentation product, right? Who first reported the external and internal mode of transmission of stewart wilt of corn? That is Pantovia stewarti. For the first time, it was reported by E.F. Smith. Sumatra disease of clove is caused by Ralstonia zygi. Which of the following involves the formation of nitrate from ammonia? This is generally nitrification. Through the process of nitrification only from ammonia is produced from nitrate, right? So proteobacteria is named after Greek god Proteus or Protus, I think so. The antibiotic acting on cell wall. So penicillin, bacitracin, cyclospora, I think all the above, they alone act as all the given antibiotics act on cell wall only. So the answer is all the above thing. Black leg of potato is caused by Pectobacterium keratovorum, right? Shape of bacterial cells before binary fission. So before binary fission, they look like a dumbbell shape, like during binary, like they look like this. A new name of Agrobacterium tumefaciens. Now it is called as Rhizobium radiobacter, based on 16 SRRNA something, but it is still a topic of discussion, right? First genetically engineered and biotechnologically produced vaccine was against. The first vaccine was biotechnologically produced and engineered. The vaccine is smallpox. Plasmid can be generally occurred in double stranded, because during definitions itself we might have discussed plasmid is a circular double stranded extra chromosomal DNA that is present in the bacterial cell, right? And for further details, student can refer this book, A Vision into Plant Pathology, complete student version. Further doubts, clarification and guidance, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.